the highest point on Earth, Mount Everest.
Là, je vais vous dire.
Là, c'est l'enfer. Non, là, c'est pas facile. Là.
On est hyper motivés, mais on serait vachement mieux ailleurs.
21h45.
She's climbed to the top of the world, one step at a time.
C'est magique, incroyable. Yeah!
An ordinary woman conquering all 14 of the world's tallest peaks in her own time and outside major competitions. She's the first French national to achieve that feat. Now she's on to her next and perhaps biggest challenge yet. Unlocking the mysteries of life on Earth, trapped within glaciers. This in the hope of reining in climate change. To her mountaineering gear, she's now added a science kit. In Peru, she collected the mission's first ice sample.
We have collected all this. We're gonna close the bag.
The goal is to establish a global database of glaciers' environmental DNA. With each ascent, Sophie Lavo is in charge of collecting 3 liters of ice at 3 different altitudes. Water teeming with microscopic life, microbes, plants, fungi, and animals frozen in time.
Okay, we gotta not stay too long here. It's too cold.
Once the ice has melted, the water is carefully filtered, a scientific protocol the mountain climber was trained to replicate. The filter, containing a high concentration of living material, is then sealed. The mission covers 6 continents, with stops in Peru, Nepal, and Kenya, among others. We meet her in Switzerland on her way to the Arola Glacier, before her journey comes to an end.
Try to let yourself slide a little. Yes, that's it. Skidding like that. We're at the foot of a block of ice from the Arola Glacier, and we're going to get some ice here. To complete the protocol, we're going to take a photo of this location and take the GPS coordinates. There you go, 2,445 meters.
A few hundred meters uphill, she takes one last sample, straight from a rock that's been in contact with the glacier.
This is not ice, it's surface material. We take a small swab and scratch the surface of the rock that isn't covered in snow.
10 kilos of samples later, the team head back to the lab. The project's glaciologists and biodiversity experts have traveled from all over the globe to meet here for the first time. Eager to find out more about the survival mechanisms developed by glacial microorganisms over thousands of years. Nature Metrics, a startup specializing in environmental DNA, is responsible for sequencing and storing the samples.
You can imagine like in a crime scene where there's DNA from humans, we do the exact same thing but for wildlife. So you can take the traces of DNA that exist, so in this case across our glaciers that are rapidly declining around the world, we can take the traces of DNA from those glaciers and then find out what wildlife is living there.
The results are then analyzed by Tom Baden's team of scientists who study the rapid environmental changes happening at high altitudes and their impact on ecosystems.
The science community is pretty much convinced that there's a huge potential contained within this glacier, and it's about time to kind of harness the power of these microorganisms.
Some species living in glaciers and invisible to the naked eye are believed to generate energy when exposed to extreme conditions, low temperatures, lack of food, and multiple freeze and thaw cycles. properties now at risk of being lost forever.
Each degree of global warming, we risk to lose our own heritage that's evolved over millions of years. We are hugely risking our own futures by not storing, keeping, documenting, and protecting that biodiversity.
Glaciers play a crucial role in regulating our climate. Now, by protecting the tiny species living within them, they may also hold the key to many of humanity's future challenges.
Well, it was Valerie de Camp, our environment editor, who worked on that report, and she's with me here
Hi, Nadia.
in the studio now. Hello, Valerie. Look, it does seem, it is unfortunate. Glaciers are vanishing. We saw that very clearly in your report there. Not just glaciers, though, that could be lost forever.
That's right, Nadia. When we hear about melting glaciers, we think about freshwater resources at risk, rising sea levels, but we don't necessarily think about biodiversity loss. And glaciers might look like sterile, frozen deserts, but they're actually full of life. Microbes, bacteria, viruses, fungi, all sorts of things. And scientists believe the biomass that it is stored within the world's glaciers is comparable to what lives in soils of all the rainforests in the world. So it's not just frozen water. But up until recently, unlike oceans or forests, perhaps, there hasn't been that much scientific interest from a biodiversity point of view when it comes to glaciers. And that is why scientists are now racing to document and preserve whatever lives in the ice before it becomes water. Now, you're wondering, why should we care about those tiny microorganisms, the bacteria, microbes living in the ice? Well, scientists have found that, we know as a matter of fact that they're the most ancient form of life on Earth, and so they can help us better understand the evolution of the planet, but they could also help us in our future. They have discovered microorganisms, for example, living in glaciers that developed a protective layer against UV radiation. They also found other species that, for example, use different sources of energy, not just solar, but also mineral and gases in order to survive. So it all sounds a little far-fetched and disconnected from our realities, but scientists are quite serious about harnessing the power of those microorganisms in order to cope with the effects of climate change.
And look, this is a mission that relies on environmental DNA, Valerie. Could it be a game changer in the fields of science and conservation?
Well, it's a very powerful tool, and it is changing the way scientists approach their work. Because eDNA, environmental DNA, traces the genetic material left behind by species, whether it's in the water, ice, or soil, you can detect the presence of species without having necessarily seen them, because you're using the DNA. And that is very powerful if you want to monitor and detect species in very remote, hard-to-reach locations. For example, it's been used to monitor endangered snow leopards, for example, which are very hard to spot in the wild. It's also being used to detect rare deep ocean species, which nobody had ever seen before. Also invasive species, so species that do not belong in a given habitat, which you don't always see with your eyes as well. So all examples of how we have used eDNA in the past. And that means scientists, for example, don't have to fly in numbers all around the globe in order to do their research. They can rely on local personnel. They can take the samples and then send the results to labs around the globe. eDNA is also very powerful at generating data on a massive scale. So with, you're able to scan for hundreds of species at a time. And that is very useful for conservation efforts, because scientists are able to prove that, a given place is a biodiversity hotspot with hundreds of species living there, which could be hard to do if you're essentially looking for every single species. And that helps them, for example, raise more money for conservation efforts. So it doesn't mean all traditional methods are not going to be used anymore, like field trips or camera traps, but it is changing the way that scientists are doing their work, definitely.
All right, really interesting. Thank you very much. Valerie de Camp, our environment editor, for us there.